Welcome back, everyone, to the second half of the American Society of Naturalists Young Investigator presentations. Our next presentation is by Matthew Pennell, and he'll be speaking on macroecology across deep time. Matt?
Thank you, Mark, for that introduction here. So this is my name, Matthew Pennell. I'm at UBC. And first of all, we're doing this. Okay, well, that's a good talk. That's all I wanted to give today anyways. Okay, there. So first, I'd like to thank the American Society of Naturalists for this wonderful, amazing prize. It's such a huge honor. Looking at the list of nominees before is just amazing. It's all my scientific heroes. And it's also an award from my peers whose research I read and whose papers inspire me. And second, I'd like to dedicate this talk to Paul Joyce, who taught me everything I know about statistics. And he died tragically a few weeks ago, so let's dedicate it to him. So I gave a nice title for my talk, but here's the real title for my talk. I don't really have, the goal of the American Society of Naturalists is to enhance the conceptual unification of the biological disciplines. And so I kind of wanted to do, instead of a research talk, to try and take up this mantle and try and give my perspective on how macroevolution and community ecology are related, where they've been, where they are now, and where I think they should go. So this talk will be very biased, it'll be very opinionated, and deeply unfair to a lot of cool research. Hope you forgive me for that. So, community ecology has been baked into the macroevolutionary cake since the very get-go. Name your favorite macroevolutionary model: adaptive radiations, escape and radiate, taxon cycles, you name it. Community ecology is kind of at the core of it. And if you go back to the paleobiology literature from the 70s, Jack Kapkowski developed the first diversity-dependent models of diversification, in which the presence of other lineages inhibited diversification in other lineages. And if you read his papers, it's very clear that he was reading MacArthur, and he was reading Wilson, and he was reading Simberloff, and he was looking to these equilibrium theories of diversity to get his ideas from. And as most of you know, this idea of diversity-dependent diversification has seeped into my discipline, phylogenetic biology. And the idea is pretty simple here, is that if we imagine that lineages are competing along a resource axis, and lineage forms or arrives at a new island and starts diversifying, then initially diversification should be pretty rapid and just slow through time. And it should leave a signature in the phylogenetic tree shape. And we see over and over and over again this kind of tree shape with lots of tiny branches at the beginning and lots of long branches towards the end. And we've interpreted this as diversity-dependent diversification or ecological limits. And we see this all the time. However, as you probably also know, there's tons and tons of processes which also can lead to this, and tons of statistical artifacts which also give rise to this. And Luke Harmon and Susan Harrison did a good job of outlining these kind of arguments in their 2014 AMNet debate, so I'm going to leave that there. But a really damning piece of evidence against the diversity-dependent interpretation is that we'd also expect it to see it in trait evolution. Now, a common thing people do with trait models is they fit alternate models and try and use the relative support for a given model to lay evidence for a claim. And if diversity dependence is actually due to niche filling, we expect this kind of early burst pattern we see on the right, where lots of evolution is fast, then it slows down. And authors often fit this to data and compare it to like an Ornstein-Uhlenbeck, which is like a single peak clade optimum kind of model, or a Brownian motion random drift model. And what we find, this is some of my data of 337 plant functional trait data sets. We find over and over again that early bursts are very, very, very seldomly supported. Most comparative data sets are kind of Ornstein-Uhlenbeck-like. And Graham Slater and I have made some arguments that maybe it's hard to detect early bursts, yada, yada, yada. But I think the overwhelming evidence is clear to me, at least, that these early bursts are rare if they exist at all. And so in this case, not only do we have not really clear interpretation. We also have very little data to support this idea. And this is a caricature, of course, but this is a figure from MacArthur and Levins from 1967. And this is kind of how they imagined ecology working, is that splitting of that species split a resource axis and then pack them as tight as they can get into a given area. And even though I realize that macroevolutionary biologists know community ecology is a lot more complicated than this, this is still kind of what our models look like and how we think about conceptually this problem. You can consider this as a bunch of animals packed in this kind of thing, and they have like a ring, and how many can you get inside there? And as Susan Harrison so nicely put it last time in her AmNet debate, we've come a long way since the folk music days of ecology, the Peter, Paul, and Mary time of ecology. So I guess my challenge for us today is, and from the other perspective, ecologists have been told that they need to incorporate macroevolution into their kind of ideas, and that paleontology and biogeography and phylogenetics have a lot to say for community ecology. This is a Bob Rickleff's quote from a 97 paper, and there's this really, really nice book by Dolph Sluter and Bob Rickleff's, which they called, if you just read through this volume, which I think is the best volume in evolutionary biology history, the best edited volume, there's so much enthusiasm for how much that phylogenetics and systematic kind of approaches can contribute to ecology. So out of this comes phylogenetic community ecology. And the basic idea, which is articulated by Cam Webb and others in the early 2000s, was basically that the effects of competition will leave a signature of over-dispersion on the phylogenetic tree. And contrastly, habitat filtering will leave a signal of phylogenetic clustering. And this paper, as I've showed you, has been cited like 1,741 times. It's a huge field in ecology. That's cool. And it's blossomed into this complete disaster. And this is from a recent survey of metrics and methods from Elliott Miller et al. And they find there's 22 alpha diversity metrics, 10 beta diversity metrics, 9 null models to test these things, and they all have one theory. Close relatives compete, competition leads to exclusion. And they can measure your tree in every which way, and it's hard to really know what's going on here. So then, of course, in this new thing in biology, pattern does not imply process, and Mayfield and Levine have this really nice paper in which they show that competition, while it can lead to over-dispersion, can also lead to phylogenetic clustering, and habitat filtering can lead to over-dispersion and clustering. We can't get the pattern. But at least we would like to see the pattern stay, but sadly that's not the case. We would predict that if it is the effect of, if phylogenetic niche diversity is important, then we should see that more phylogenetic diverse assemblages are occupying a broader niche space than other assemblages, and that's actually not the case. It doesn't seem to be the case at this point. So there's a lot of talk about the dark side of comparative methods. I was in a symposium a few years ago in which we talked a lot about statistical problems in comparative methods. And you may have gone to a lot of talks this week where people told you about different priors or different detectability problems or whatever. And I think stats are extremely important. Stats are awesome. I love them. I care about them. But I think the real dark side of comparative methods is that we often don't know what we're measuring or how it relates to what we care about. And so this is the central question posed by my talk. Can we do better? And my answer is a caveated, I think so. And so one thing we could do is to just blow it all up and start over again. And I think this is the neutral theory of community ecology is really cool for this, because it's like an attempt to reimagine how community ecology and macroevolution could be joined together from the get-go. And say what you will about neutral theory, at least it's the benefit of it being cohesive across scales. This is a really cool paper by James Rosenthal. Or, I think maybe more promisingly, we could update our concept of coexistence. And I think modern coexistence theory is defined by like 2 central concepts. One is stabilizing differences, and one is fitness differences. Stabilizing differences is where intraspecific competition is more important than interspecific competition, and this facilitates coexistence. And and fitness differences are where intraspecific competition is more important than interspecific competition, and this creates competitive exclusion. And so Rachel Germain has this fantastic, fantastic paper where she shows that the outcome of these phylogenetic community ecology models depend not only on what kind of fitness you're looking at, but also the biogeographic history of these lineages. And if you get nothing else out of my talk today, I want you to go check out Rachel's paper because it's fucking cool. Oh, sorry. Okay, next. You could modernize our phylogenetic comparative methods. And Scott Newsmere and Luke Harmon have some really cool work in this space. And JP Drury has an awesome paper that just came out looking at how to estimate more complex relationships from phylogenetic trees. I think this is great work. But all of this work kind of has the same kind of idea that competition is really the important thing we should be looking at, or species one-on-one species interactions. And I think there's a lot bigger scope of things to do. So in this talk, I'm going to try and lay down what I think could be a different direction for this field. So the fundamental premise I'm working from is that the scaling of biological processes is a foundational concept in modern ecology. And what I mean by this is, I'll give you some examples. There's a lot of really, really cool work in ecology these days about how functional responses vary with body size, the handling time, all these kind of things are really predictably, you can really easily predict them from body size, and I think that's cool. And this touches on what Mark was talking about in his AMNAT address. There's some really neat theoretical work and empirical work showing how trophic cascades and whether they happen or not depend on body size, and these all scale together really nicely. And there's some like weirdo things in ecology which seem like hard to explain, but this is a reaction norm that basically if you put something in warmer temperature, it gets smaller, and it gets smaller proportionally. The bigger things get smaller, bigger things get more proportionally smaller. And so there's all the kind of scaling relationships which really are important for understanding community ecology. Now, a really, really important one that has been touched on a lot is this kind of relationship between metabolic rate and body mass. This is a linear relationship on a log scale, and basically what it means is that, okay, so the scaling coefficient is usually less than 1, which means that larger organisms proportionally have lower metabolic rates than smaller organisms. And there's some really nice, amazing theory which shows that under certain fractal assumptions, you get this kind of relationship equals 3/4, this quarter power scaling, which is the metabolic theory. And quite frankly, I don't really care too much about this fractal thing. It's cool, but like it's not my interest here. But the point is that with this fractal without, it's the linchpin of this kind of metabolic theory of ecology. And you can use this kind of value of theta, the relationship between body mass and metabolic rate, to make predictions about the responsive physiology of temperature changes, to biomass production, to individual growth rates, to population parameters, and the distribution of lineages and traits across space. This is a very, very powerful framework. And it's really cool. And some of my favorite papers that I've read in the past little bit have been looking at using this kind of metabolic theory of ecology framework to make predictions of community structure. There's this conceptual framework on the left here by Ben Gilbert et al., where they looked at stability of consumer resource models under various temperature regimes, and they built this all off of metabolic theory of ecology. And this really, really, really nice paper by Bernacchi et al. In Ecology Letters a few years ago, in which they used metabolic theory of ecology and first principles to make really, really nice predictions about the distribution of biomass in response to primary productivity and temperature in a bunch of reef communities of fishes. And it's really cool that this metabolic framework is so predictive of so many things. So here's where my project comes in. This is a joint project I'm going to talk about with Josef Weda, who's the real MVP of this show, and Rafael Maya, Elliot Miller, and Craig McLean. And what we wanted to ask was, how does this relationship evolve? So we have this simple linear relationship that's important for all these ecological variables, but when people have examined this, they assume it's like a static constant, like a physical constraint. And we want to ask, is there a way to think about this as an evolving trait? And so I just wanted to take a brief detour and talk to you a bit about review allometries here, so you know what I'm talking about. There's three main types of allometries. There's ontogenetic allometries, in which the same individuals are sampled at different times, and this reflects development. We have static allometries, which are different, same lineage in different individuals, and talk about across the population scaling. And we have the so-called evolutionary allometry, which is the different lineages, are the points of different lineages, and the points represent lineage means. So right here, this metabolic scaling relationship is actually an evolutionary allometry. But this brings up the question, how can we have an evolutionary allometry without any evolution? As I told you before, this is assumed to be a constant throughout time and history. But this is actually a tricky problem, because I want to pose the problem is, how can we study the evolution of a trait that we can't measure? And why we can't measure it is because in any given species, the allometry is only defined within relation to all the other species in the group. So in any given species, we don't actually have information on the general allometry. So this is a really tricky problem, and I think this is maybe why it hasn't been studied very often. But my basic argument here is that we have the conceptual tools in macroevolution to think about this. And that's this idea of the macroevolutionary landscape. We're used to thinking about fitness landscapes all the time, of like fitness peaks within populations. But a macroevolutionary landscape describes the structure of population fitness landscapes through space and through time. This is a very old idea. It dates back to Simpson back in the 40s. And he imagined that niche space was carved up into these adaptive zones, and that lineages can move between them at different times. And I think this is also really what we're measuring when we're creating these kind of multi-optimum Ornstein-Uhlenbeck models. This is a really nice paper by Luke Mahler and friends, in which they use these multi-peak OU models to try and describe the adaptive landscape of the anolis lizards. And they found evidence of convergence and this kind of stuff. So we kind of took that idea that, hey, we can borrow this concept from macroevolution and we can maybe start studying this. So we basically derived a new form of an Ornstein-Uhlenbeck model. And normally when we talk about Ornstein-Uhlenbeck models, we're talking about the evolution of a single trait, and we're following its dynamics across the tree. But in this case, we're having this dual process where these lineages are evolving around an optimum OU process, and the optimum itself is also evolving across the tree. And so what I mean by a very adaptive landscape is that basically the species data points we're measuring, the body metabolic rate and the body mass we're measuring, are basically just like shadows in Plato's cave or something like that. We're interested in the actual general structure, and they just give us hints as to what it looks like. You can talk to us about the math and stuff like that later, but it's basically a linear formalization of the OU process. So then we also combine it with this statistical trick called reversible jump Markov chain Monte Carlo, which has different proposal mechanisms which can allow parameter space to grow via split proposals, combined via merge proposals. And the point of using this is we want to detect transition points in the OU regimes. And it also allows us to compare models with different predictors using Bayes factors. So we use mostly old data here. I didn't put any animals in like a thermal chain, I don't even know what that means. But we went and collected from the literature 857 species of vertebrates, mostly from a paper by White et al. And some others. We cleaned it, we standardized it. It was a lot of work. And then we went to the open tree of life and started collecting phylogenies for all these different groups, combined them in hopefully useful ways, so we could have a big tree and study the evolution of these traits. So here's what the data looks like. We have log body mass on the X and metabolic rate on the Y. And you can see there's some kind of clustering here, but we're not really sure if that clustering is related to phylogeny or what that means. And so what we did is we ran our reversible jump MCMC OU process across this tree. And the colors represent some different variation in the parameter values we get here. So on the left, we have the intercept. On the right, we have the slope of this relationship. And as you can see, there's quite a bit of variation across this tree. And we also see some really good evidence for some distinct shifts. So I think there was 8 shifts that were really, really well supported on this tree. You can see the groups on the bottom there. And interestingly, these shifts, even though we let the MCMC machinery put their shifts anywhere they wanted, they often ended up at the base of major clades or major transitions. So we thought that was kind of curious. That was interesting. But I think a bigger, broader, more conceptual answer we got was this kind of what the structure of this adaptive landscape looks like. And so we found that transitions between adaptive zones are rare. And so there's only 8 shifts across this whole tree that led to clades of more than 5 taxa. And so, and these are often associated with major clades or transitions. An example is the plethodons have a unique metabolic parameters, and they're the lungless salamanders, which are doing things very metabolically different than other things. But what's really interesting is within each adaptive zone, evolution is highly constrained. That is to say that there's lots of phylogenetic signal between major groups, but little phylogenetic signal within major groups. And so we were really interested in understanding, like, what could have caused this? So we tried a few things. None of them really worked that well, but it was okay. So first thing we tried was genome size. And this is often used as a proxy for cell size. And we might think, based on the metabolic scaling arguments, that cell size is going to be super important in this relationship. Turns out it matters a bit, but not that much. Another kind of tantalizing idea that we had was that maybe instead of having different linear parts, we actually have a curved relationship. And this was suggested from this theory by Kolkatronis et al. In Nature. And so we tested these models as well. And they appear to matter in some clades, but are definitely not a general explanation for the shifts and the general pattern we observe. So to sum up, we have this interesting pattern of rare shifts and tight constraints within these shifts, but we don't really know what's driving it. So what's next? First, as I hinted at, I think we really understand what processes are causing variation in these traits. This is our analysis. It's a limited scope to some mammals or some vertebrates, but I think this kind of thing can be expanded a lot, and we can identify a lot more clades which do things differently, and then try and find ecological and evolutionary causes to generate these kind of variation. I think that's a really interesting program. And I think what we've clearly demonstrated here that this is not a physical trait. It's not a physically or a law of physics. It's something that's controlled by evolutionary ecology. And I think people in here have lots of good ideas about that. But I think what's really exciting to me is that how are the predictions from scaling relationships change if we consider that this is an evolving trait? I showed you these papers by Gilbert and Bernacchi, and these have all assumed a static relationship across lineages, but what if we start tweaking this? And what if we start using our parameter values we've obtained from our MCMC run to try and get at how this affects our predictions, and does it matter, and this kind of stuff. And there's a bunch of macroecological theory, from metabolic scaling to the more recent maximum entropy theory, which all depends on these kind of values. I think we can make some interesting predictions about this. And as I mentioned before, I think scaling is a dominant theme in modern community ecology. I think the framework of thinking about these scaling as being an evolving trait in this adaptive landscape kind of way, I think is a really powerful way of thinking about the thing. And we can start looking at how functional responses have changed over time, how responses to temperature have changed over time, and how trophic cascades may have changed over time. And I think what I really want to get out of this is I want to learn how constrained these processes, these things have been throughout evolutionary time. And what can we say about the system that's existing today? Was it always like this, or could it have been different? And there's this really, really cool part of literature in paleontology, which is probably not touched on by anybody else in macroevolution. And what these people have done, starting from Seth Finnegan has a nice paper on this, and then goes back to Baumbach in '93, is trying to use the abundances of different lineages and as well as their body masses to predict whether energy is fluctuating through time and how it's fluctuating. This is kind of a weirdo question I've always wanted to answer, which is, are things getting more energy efficient towards the present, or are we kind of in a steady state? And in both these papers, they've assumed that basically the scaling coefficient is constant for time, but what if we thought of it as evolving? We can start generating models of macroevolution that link linked up to macroecology and ask questions about how energies change through time. And a big thing I would like to say is we also want to synthesize comparative and with other types of data, such as field and experiment data. Chris Muir has this fantastic paper, which if you also read Rachel's paper and still looking for something awesome to read this paper because it's great. Is what they did was they measured the static allometry in a bunch of different species of tomato plants and then modeled the evolution of this static allometry, which is a bit different than what we're doing. But how these different allometries are related to each other is super interesting from an evolutionary ecology perspective, as well as from understanding the ecology of these communities as well. So I think this is a really, really promising thing that we should be working on more. So to sum up my rant here, is that the scaling of biological processes connects organismal to community to ecosystem ecology. I think this is a very, very powerful idea in biology. And we haven't basically, as macroevolutionary biologists and phylogenetic biologists, touched on this idea at all. But we already have the toolkits, both conceptually and in large part statistically, to start thinking about how to do this. So we can borrow from all the great work that all these people have done in phylogenetics and macroevolution to start thinking about how these traits evolve. And I think also I want to learn about the constraints that have shaped and will shape higher levels of organization. I think we can have like a predictive macroevolution, which we can start using macroevolutionary results to inform about how climate change and these other kind of things will affect our ecosystems. And you may be wondering right now if that I'm doing the Underpants Gnome scientific method. Phase 1 is understand long-term dynamics of ecological scaling. Phase 2 is question mark. And phase 3 is unite macroevolution and macroecological research. I admit this is kind of true, but I'm working on phase 2. I think this is going to be a huge area of research. And I think the approach that I've suggested, the ideas I've suggested are getting at the history and the future of ecosystems in a way that other types of macroevolutionary research is not. I think there's tons of room for community ecology and macroevolution to share ideas back and forth. And I think it's different than what we've been doing in the past. So with that, I would just like to throw an advertisement. I'm starting a computational biodiversity research group at University of British Columbia. And if you like what you've seen here and want to come help me do it, come talk to me. If you don't like what I've done here and want to help me fix it, come talk to me. Or if you're just a cool person, come talk to me. And with that, I'll just end it, and thank you very much. And also, I'm having trouble hearing today, so if you have a question, just please come talk to me in person. I probably won't be able to hear you, so thank you very much.